Medtronic Technologies impacted more than 72 million people in the last year, equating to 2 people every second. Harnessing the power of technology to take healthcare further, each technology has unique benefits designed to serve patients. The goal of this program is to get closer to the patient and delve into the challenges and impact of each technology in practice. This is the Medtronic MedEd Learning Experience. The Nellcor pulse oximetry monitoring system should not be used as the sole basis for diagnosis or therapy and is intended only as an adjunct in patient assessment. Medtronic's medical education programs are offered to provide attendees education on the FDA-cleared indications and use of our products when applicable. The contents and conclusions of the following program are solely those of the speakers unless otherwise cited. The speakers are responsible for all content and any necessary permissions. The speakers received funding from Covidien LP, a Medtronic company, for this speaking engagement. For this introduction segment of the series, a discussion on the Nellcor technology. We will discuss pulse oximetry and the impact of skin pigmentation on the technology. To help provide insight into this topic is Jake Dove, Senior Principal R&D Engineer at Medtronic.
So as we dive into this, we like to think of a pulse oximeter as having 3 main components: a sensor, electronics, and an algorithm. And what we mean by those components of the sensor, that is the probe that's going to have the light. It's going to be able to deliver and collect light from that patient. The electronics will measure that optical signal and convert that signal into something then that the algorithm can use to be able to A, locate the arterial pulse, and B, calculate pulse rate and SpO2. And what we want to talk about today is how do we go about engineering a system so that it can work on all types of patients. And the first thing that we have to do is we have to understand, well, what are the types of signals that we will expect? And throughout Nellcor's history, we have developed a signal space for pulse oximetry. That signal space we can think of as varying amounts of light that's collected, shown here on this horizontal axis, versus on the vertical axis, how much blood signal do we have, or what is the percent modulation of that detected signal? So here we can look at 2 different cases. We can examine 2 different cases. So one, we could have a patient that has an okay blood signal and a lot of light that we're collecting. What would that patient look like? Well, they're probably fairly healthy. Because we're getting a lot of light, we would say that they're probably on the lighter pigmentation side, and that the sensor site is not very thick. And so we might think a pediatric finger in this instance, so a thin sensor site. And we can contrast that, I say, with maybe an adult, so where we're still getting a good blood signal, but now our light levels have decreased a lot. And so here, when we go to the adult, what's happened? The finger size has increased, so the amount the light has to travel through the tissue has increased significantly, as well as the melanin. Having more melanin, there's going to be more absorption of that light. And so both of these things are going to decrease the amount of light that we can measure with our sensor. And so that decreases the light levels that we can detect. Now what we need to do with Nellcor, with our pulse oximeter, is ensure that we can work across this broad spectrum of light levels. And so with Nellcor, one of the things that we get to utilize are decades of data that has been collected on people. And here we can use that data to understand what the signal space is and ensure that when we make a pulse oximeter, that we're able to optimize that signal, and that we can test to ensure that our product is going to behave or operate as expected. So the first part that I want to talk about is the light level. Light level is the amount of light that you're detecting. So for sites that have a lot of attenuation, you get little amount of detected light. sites that have low attenuation, you have a lot of light that you can detect. And in those situations, the signal that we can pick up and send with our sensor will change a lot. Now, when we think of this problem, we have two levers to manipulate in the design. One is the LEDs that we use to generate the light that we input into the patient. The amount of light that an LED generates depends on the amount of current we send to that LED. So the more current we send to the LED, the more light that is generated. Secondly, the amount of light that we detect gets converted into a photocurrent that then is amplified. That amplification happens in multiple stages. So we're able to amplify that signal a little bit, or we're able to amplify that signal a lot. Deciding how much amplification and current that we want to drive that LED at is what we'll talk about here. And in Nellcor, we have developed a system to automatically do this. And so here, what we have are a couple of different cases. First, we have that case where the light level that we detect is very, very low. It is so low, in fact, that the signal that we're measuring is what we would call below the noise floor. That signal is hidden in all this noise. We can't see it with our electronics. What we need to do is to take that signal and increase it into that optimal signal range. And we can do that by increasing the amount of light that the LED emits. So here, we would want to increase the current that we send to the LED, which would then generate more light and allow that signal to be distinguishable from the noise. The second case that we can have is when the light signal that we detect is so high that it actually saturates our electronics. Now, what we mean by that saturate our electronics is that as more light is detected, our electronics can no longer measure that increase. They've maxed out the measurement capability. And so in this situation, what we need to do is to take that light signal and decrease it. We need to decrease that signal back into our optimal signal range. So there are two ways that we can get that signal to decrease into our range. One, we could send less current to the LED, or we could amplify that current less. In Nellcor, there is an algorithm that automatically adjusts the amount of light or the amount of current that we send to the LED, as well as how much amplification do we want to apply to that detector signal. And in this, every time you put a Nellcor sensor on, it runs through that optimization algorithm to ensure that you're getting the best signal possible. Second, we can go into the blood signal. So the blood signal or percent modulation. Here we have two different cases. So one, we can go back to our pediatric subject. Now in this situation, maybe their hands are really warm and they're healthy. They're really well perfused and they have a really large blood signal. So the amount of light that's interacting with blood versus the rest of the tissue is really high. So really high percent modulation. Now let's contrast it with maybe an adult, thick fingers, cold hands, or maybe on vasoactive drugs. And what that causes is a low perfusion situation where the amount of light that interacts with blood is very low relative to the rest of the tissue. And so in that situation, we have a low percent modulation. Nellcor needs to be able to work over all these ranges. The low light level, low percent modulation is one of the more challenging conditions for a pulse oximeter to be in. And for a pulse oximeter to work well, we have to go back to our system level thinking of that pulse ox, and everything has to be designed to be able to deliver that performance, and it starts with the sensor. And so here at Nellcor sensors, I like to show this example because in Nellcor, Nellcore is rated down to a percent modulation of 0.03%. And here what we're showing is stepping down from 5 to 0.5 to 0.05 in that plot in the lower left-hand corner. You can see comparing 0.05% modulation to 5% modulation is tiny. A little bit of noise that gets on that signal will wreak havoc in the processing later on. And so a lot of care has gone into designing our electronics and our sensors to block any electrical noise. And we can see this with the Nellcore sensor, where we have the sensor cable. If we cut that cable open, we'll expose an outer electrical shield. That outer electrical shield, if we unravel that, we'll find two conductors. Those conductors, if we trace them over, will be attached to the LEDs. And if we look there, we find another inner jacket. And if we cut that inner jacket open, what we see is that there's a second or an inner electrical shield. And that inner electrical shield covers the photodiode conductors. Now, the photodiode or the detector is also covered with something that's been specially designed to block electrical noise but allow light to pass through it, and that's called a Faraday shield. And so here we can see that is this piece of copper that folds over our detector when in use. And in this situation, now what we have when this is folded over, we have 100% coverage, or we have maximum coverage of electrical noise on inner cabling and on our signals that we're trying to measure. And it's in this way that Nellcore is able to go all the way down to 0.03% modulation. And I like to really kind of show the sensor is one piece of this, but everything has to work together because 0.03% modulation is really small. It is like trying to find a blade of grass on top of the Empire State Building. And so if you think of it, our signals that are incoming are like the Empire State Building. And what we want to find when it's 0.03 is this blade of grass. And so we have to have everything working in unison. It has to be working well designed so that our device can perform as expected. One of the ways that we ensure our products do that is we go back to that signal space that we had talked about. And in that signal space, we will test our pulse oximeters. And we can test them. One way to do it is with a Fluke biosimulator that has an SpO2 functional tester. What this does is it generates optical signals that match certain light levels and certain percent modulations. At those optical signals, we can look at our device and we can ensure that it's performing as expected. And so in this situation, it's a big test where we change SpO2 and heart rate and the light level and percent modulation. And we can test all of these different conditions. And what we can find, what we want to see, is that no matter where you're at in that test condition, the Nellcore pulse oximeter is performing very well, and that's what you're seeing here. This is important for both the design as well as the support of released products. So once our pulse oximeter goes out into the market, we want to make sure that you continue to have that high quality performance you've come to expect. And so as we support devices throughout their life cycle, components might need to be updated. Will the new components meet the same performance specifications as the old components? And we can test in these situations to ensure that they will. Lastly, we can also see this play out where we specifically within Nellcore looked at validation data over different pigmentation subgroups. And when we broke out all of our validation data into light pigmentation and dark pigmentation, and then we analyzed that performance, that error, so the SpO2 minus the SaO2, so the arterial oxygenation reference. When we look at that, what we found is that for light and dark, they both met our accuracy specifications of less than 2 RMSD. and that's between 70 and 100% saturation. And so what we found when we broke out these two groups and we analyzed their results is that between 70 and 100% saturation, both groups met that RMSD of 2 that Nellcor claims. We did notice that there was a small increase in bias in the darker pigmentation, and we did notice that there was a small increase in the bias for the darker pigmented subgroup, and that is something that we're actively examining in Nellcor. How can we make Nellcor perform better on all pigmentation groups?
Please tune in next week for a new segment from this series wherever you find your podcasts. This is the Medtronic MedEd Learning Experience. Thank you for listening.